It is day one of the JP Morgan Healthcare Conference, and I am delighted to be joined in the flagship studio by Mike Severino for the fourth time since you and I have been doing this. Welcome, Mike.
Pleasure to be here.
So, Mike, Tessera has developed a number of programs with its gene writing technology. I want to focus on two today, one being AATD and the other being sickle cell. Let's start with AATD, because you had some exciting news this morning where you announced that you have regulatory clearance to start clinical trials. Congratulations.
Thank you. It's an important milestone.
Yep.
Maybe just a minute about what alpha-1 antitrypsin deficiency is.
Please.
It is a rare genetic disease by regulatory definition, but it's not that rare. There are about 200 to 250,000 patients in the US and Europe who suffer from this condition. And all of the patients have two copies of the same mutation that causes them to make an abnormal protein. And the consequence of that abnormal protein is they get a destructive lung disease and they get a progressive liver disease, both of which can be debilitating.
So two organ systems.
Two organ systems affected by the same mutation, which unfortunately can be debilitating and life-shortening. So we have a therapy that can correct that mutation back to its intended sequence, what geneticists would call wild type, the normal sequence that exists in you and me. And we've advanced that therapy. We've generated some tremendous results preclinically. We can correct approximately 95% of the alleles in model systems that cause this disease. And this is an autosomal recessive condition, which means 50% is probably enough to cure the disease, and we're in the 90s. So we've advanced that through preclinical studies. We filed regulatory applications around the world, and we're fully approved to start clinical studies. In some important geographies, we have all the approvals we need in Australia. And we just announced this morning that our US IND has cleared its review, meaning that we can initiate studies in the United States as well.
Terrific.
That's an important milestone because US regulators tend to be the toughest, particularly for genetic medicines. And the fact that we cleared our IND hurdle without any significant setbacks and got it right the first time, I think is a testament to the quality of both the preclinical program that we've designed and the candidate that's going to be going into the clinic.
So I want to talk about that moment from two different perspectives. One, why is AATD the right place for Tessera to start? What's its gene writing technology? And then I'll come back to my second thought after that.
Well, it's a condition that meets all the requirements that we have when we think about indication selection. So it's a severe illness. It's debilitating and life-shortening. And we're treating severe illnesses, particularly early on in our journey as a company. It's an illness where, unfortunately, the existing standard of care is woefully inadequate. The only thing that can be done today for these patients beyond symptomatic treatment is to try to give them replacement enzyme from healthy donors. That has only marginal impact on the lung disease, and it has no effect whatsoever on the liver disease. So there's a real gap in therapy for these patients. And it's caused by a single mutation expressed in the liver. So it's a tissue that we can access with our technology. And it's something that we can correct with very high efficiency. So when we look at all those factors together, they make it an ideal place to start our journey as a therapeutics company.
Okay, great. So that same decision, but looking at it through the industry lens, as I understand it, this is the first regulatory approval of an in vivo genetic editing technology of this kind. What should the industry take away from this decision, this moment? What should we be looking for as this therapy progresses?
Well, I think it's an important milestone not only for us, but for the field. It's the first time a construct that uses the mechanism that we're using to modify the human genome has been approved for clinical studies. That's an important milestone. We'll be entering the clinic, actually enrolling patients in the very near term. And before long, we would expect to have human clinical data. And that is really the ultimate proof. You know, we can do tremendous things in model systems, in engineered mice, in non-human primates. Those are important predictors of success, but there's nothing like having the human clinical data, patient data, showing that a patient who's suffered from this genetic condition for his or her entire life can now make the normal protein because we've restored their genetic sequence to the intended sequence, the normal sequence, that can give them a long and healthy life. And that's an incredibly remarkable journey to be on. Yeah. It's been many years since the founding of this company to get us to this point, and we're just a very short period away from having that sort of clinical data, and it's a really exciting time at Tessera.
to reference Newbar's letter from this morning, that sounds like a miracle in the making for sure.
It is. The sorts of changes we can make today, we couldn't even imagine when I started in medicine 25 years ago.
Yep. And that medicine has attracted the interest of a partner in the form of Regeneron.
It has.
Tell us about what that partnership enables you as Tessera to do.
So it's a global 50-50 collaboration. So we'll share in development and commercialization as 50-50 partners. We're going to be leading the program through clinical proof of concept. The partnership does a lot for us. The partnership gives us breadth and scope and scale in late-stage development that would be difficult for us to build first time through. And it gives us that same scope and scale for commercialization, which will be important.
Great.
Regeneron, I think, is an ideal partner for us. They're a very well-regarded, very rigorous scientific partner. We are very rigorous scientists at Tessera, and I think there was a very early meeting of the minds. And I think they, like we, feel like this candidate has just tremendous possibilities and great potential to be a curative therapy for patients where none exists today.
And they've been a pioneer in the genetic medicine space for a long time.
They are. They've been a pioneer in many fields.
Yep.
Antibody development, novel therapeutics, but they've made a real commitment to genetic medicine and have significant expertise and have made significant investment there. And so I think it's nice validation of the work that we've done to form this partnership. And as I said, I think we saw a very strong scientific and cultural fit from the beginning of our discussions with Regeneron.
Very nice. So let's switch gears and talk about sickle cell. Describe for us a little bit about what that program is and how it's different from others that are also working in the same space.
So our sickle cell program is a very interesting program. What we've done is we've taken our delivery technology, which we developed in-house, which allowed us to correct in liver the A1AT mutation, as I just described, and we've essentially reprogrammed those LNPs to go to a different tissue. And it's a very challenging thing to do. It took a couple of years of work in our labs, but we've achieved breakthrough results that allow us to detarget the liver and direct our editing constructs to the long-term stem cells in the bone marrow that make our red and white blood cell systems.
So same editor, different tissue.
Similar editor, different tissue.
Okay.
We've developed an editor that can change the sickle mutation to normal, to, again, what geneticists would call wild type. Sickle cell disease is caused by, again, a single point mutation. So one base pair wrong out of 3 billion. And if you get 2 copies of that, one from your mother and one from your father, unfortunately you'll have sickle cell disease. We've known that for 75 years. But there's been nothing that we can do about it until very, very recently. There are ex vivo therapies, gene therapies for sickle cell disease, but they require a stem cell transplant to administer. So fully myeloablative, and what I mean by fully myeloablative is completely destroying the normal marrow with old school chemotherapy, the same kind of chemotherapy that was used in the 1950s and 1960s, and then rescuing the patients with modified cells in what is essentially an autologous stem cell transplant.
Yeah, very
it
difficult.
Is a wonderful thing to cure any patient of sickle cell disease, but very few patients with sickle cell disease can go through that kind of treatment. Most aren't considered candidates, and there are also significant limitations in health resource utilization. There's only so many transplants any given center can do in a year, and they have a wide range of conditions that they need to treat with stem cell transplants. So while those therapies are a great scientific achievement, as a practical matter, it'll be very difficult to treat anywhere near the 150,000 patients living with sickle cell disease in the US and Europe, 250,000, broadly speaking, in countries with developed medical systems, and 6 or 7 million patients around the world. Many of those live in underserved regions like Equatorial Africa. And so we're exploring a very, very different approach. We're doing this all in vivo. So by reprogramming our lipid nanoparticle delivery systems, we're able to get to those stem cells in the marrow, in their native niche, where they live, where they do their job, without the need for stem cell transplant or any of the manipulations that I described before. And we can rewrite with high efficiency from the sickle mutation to wild type in a number of model systems. And that program is not far behind our alpha-1 antitrypsin deficiency program. So, you know, as we go through all those important milestones that I described for alpha-1 antitrypsin, on the back end of that, we'll be going through that same sequence with sickle cell disease. And we believe we stand an excellent chance of bringing forward the first curative therapy in vivo for sickle cell disease, which would be a phenomenal accomplishment. And this is a kind of therapy that can not only be used in the developed world, but we believe it can also be used in underserved regions like Equatorial Africa and other equatorial regions. And the Gates Foundation agrees with us on that. They have a significant partnership with us specifically aimed at making sure therapies like ours get to patients in underserved regions so that they can benefit from them.
So that sounds to me like two potential miracles in the making and a very exciting year ahead for you. Tell us, as you are here at JP Morgan, you've been to this conference many times before, what is it about this year or this moment for you that feels exciting?
Well, I'm happy it's not raining. That's perhaps the first part. But you know, along with the sunny skies, I think there's a positive attitude at this JPM.
Great.
I think there's a sense that biotech is sort of getting its legs back. You know, folks are now looking forward to what the next great breakthroughs will be. And I think Tessera has a good chance of delivering two and more, because there's much more we can do with this platform beyond Alpha-1 and sickle that we had time to talk about today.
Well, thank you, Mike, for giving us your time and thoughts this afternoon. Always a pleasure to have you here.
A pleasure to be here. Thanks very much.